this on? Yeah, all right. So 2 new antibiotics is a hard act to follow, but let's see if I can bring some interest in my work. All right, okay, good. So I'm at the Indian Institute of Science, Education and Research in Pune. That's the ISER in Pune. And I'm interested largely in understanding the mechanisms of antimicrobial resistance, but also in a sort of eco-evolutionary context, because I think that we can't really understand AMR without thinking about the evolution of AMR and the ecologies in which AMR evolves. Just to show you a nice pretty picture, that's the ISER Pune main building. That second window there is where my lab is physically located, but we also have a presence on the internet, so look me up, write to me if you feel that there's something interesting going on here. All right, good. So I think we'll all agree that AMR is essentially a product of evolution, very similar to natural selection. And of course, one can't really understand evolution without understanding the ecology in which this evolutionary process is occurring. As we've said through the day, and we're going to say it again tomorrow and day after, antibiotics are everywhere, and bacteria interact with antibiotics at multiple levels. And depending on the environmental situation in which this is happening, the way that the antibiotic, or the way that the bacterium experiences the antibiotic is likely to be very different. So for example, in patient bodies, we're going to see temporal variations in antibiotic concentrations. In environmental reservoirs of bacteria, again, we're going to probably see low concentrations of antibiotics that are going to vary over much longer time scales, right? And all of these different ways in which bacteria are going to experience antibiotic is essentially going to apply different kinds of selection pressures on the mutants that could potentially confer resistance in these bugs, right? And so what we're doing is we're using laboratory evolution, in vitro evolution, using fairly well-characterized bugs like E. coli to try and understand how, when we change the parameters of selection, you change the kinds of mutations and therefore the kinds of mechanisms of drug resistance that evolve. And that's sort of the large question that we're asking. What I will do is, instead of showing you a lot of data, I'm going to just walk you through the sort of broad arcs of questions that we ask within this realm. And then I'm going to throw up a few questions in response to the questions that I was sent to kind of just make things interesting, I hope. All right, so the first kind of thing that we're interested in doing is understanding the relationship between selection pressure, and in this case, we're looking primarily at drug concentration and mechanisms of AMR. And by mechanisms of AMR, we essentially, of course, use genome sequencing to identify mutations and then infer mechanisms based on that. So we know that antibiotics, all the way from extremely low concentrations, much below what we would define as the minimum inhibitory concentration, to way beyond the minimum inhibitory concentration, can potentially enrich drug-resistant bacteria. The question, of course, is, do you get the same kinds of mutations along this entire spectrum of antibiotic concentrations? We find that that does not happen. In fact, we've spent a lot of time looking at concentrations that are about 5 or 10 times lower than the MIC. And what we find is that there are a lot of unique adaptations that occurred in this selection regime. I'm highlighting 2 of them which we've already published, and so you can go and look at these papers and get more details if you'd like. The first is mutations which change the lifestyle of the bacterium from being primarily planktonic to being pellicle and biofilm-like, get selected at low antibiotic concentrations, and this confers a sort of generalist benefit to the bacterium. So the bacterium experiences rifampicin, but because of this lifestyle change, it now starts becoming low-level resistant to a bunch of other different antibiotics, and this could potentially change, further trajectories of evolution is what we've claimed, what is our claim. And we've also shown that there are a number of transcriptional rewiring events which are triggered by mutations, which are selected again in these low drug regimes. What these will do is typically lead to overexpression of the drug target. For example, there was a great example about this mediated by small RNAs. We find that these mutations typically occur in 2-component signaling pathways, in regulators of these pathways. And in a sort of rather bizarre twist of events, what one finds is that you very often see preemptive upregulation of the drug target, which essentially then synergizes with subsequent mutations that occur in the drug target to give you extremely high-level resistance. We've also shown that if you can prevent this initial mutation from occurring, you can slow down the rate at which resistance will evolve, and also you can slow down the MICs that resistant bacteria will have. And so obviously, while these are mutations that get selected at low concentrations, they have implications for how resistance finally will evolve in a clinical setting. The second kind of question that we're interested in asking is in understanding what the consequences of trade-offs are. So we know, for example, that resistant mutations can very often collaterally change other properties of bacterial cells. The most common example is the fitness cost of resistance, which all of us have actually seen in the lab. And so what we're interested in asking is, what is the mechanism of these kinds of trade-offs, and can we potentially harness this information to design dosage regimes, for example, of antibiotics that could potentially slow down rates at which these mutations are fixed in populations. One of the very interesting trade-offs that we see is with the stability of the drug target. So typically drug targets will mutate to prevent drug binding, and again, this is one of the most common mechanisms of resistance that we see. But very often these mutations destabilize the protein, and the drug target becomes susceptible to proteolysis in the bacterial cell. So what that means is that while the protein has become resistant, its half-time in the cell has gone down. And so what we now know is that you can manipulate the proteostatic environment of the cell, the proteases, chaperones, etc., and you can actually slow down the rate at which these mutations get fixed in populations simply by exploiting this property of evolution, which is the trade-off, right? The third kind of question that we're interested in asking, and this is ongoing work, so I have no published data here, so I'm happy to talk to you later about it, is can we take drug-resistant bacteria and can we resensitize them? So what I mean by this is, essentially, can we push MICs back to the left of the breakpoint, which is really clinically all that you're interested in doing, right? And of course, we know that you can do this using very specific adjuvants like clavulinate in the case of amoxicillin. This has been a really major success story as far as beta-lactams is concerned. But we're really more interested in understanding how intrinsic resistance, or molecules that, or proteins that regulate intrinsic resistance in bacteria, could potentially become targets for adjuvant therapies. What we're doing is a very simple experiment here. We start with gene knockouts in E. coli. We screen them for hypersensitive mutants, right? Not mutants that are more resistant than the wild type, but mutants that show lower resistance levels than the wild type. And then we simply bring them into resistant backgrounds and ask, can we reduce resistant levels and bring them to the left of the breakpoint? We don't stop here though, because we love evolution. So what we do is that we take these sensitized strains and we also evolve them in the drug, and we ask how stable are these kinds of sensitizations, because finally, we can introduce any number of antibiotics and any number of adjuvants, but if the lifetime of any new compound is about a year or two in the clinic, it doesn't really help us in any major way, right? And so we're interested in kind of developing pipelines to be able to define how stable sensitization to antibiotics is likely to be, and of course we use lab evolution to do this. Okay, so I have 4 minutes before the birds start chirping, or maybe I have 2 minutes before the birds start chirping, so let me just quickly run through what I wanted to say. So obviously, we all agree that AMR is not a single problem. It's probably a bunch of problems that are linked together by very fundamental processes at the level of the bacterium, but also by human behavior, human interactions, how societies behave, how societies don't behave. And so essentially, when we're thinking about AMR, we can't be thinking about a single solution. It has to be multiple solutions, which brings all of us together because we all work in our own individual domains and try to achieve these goals. In my opinion, basic microbiology, or fundamental science, has the potential to really be the place where all of these different kinds of areas can come together, because unlike, for example, industry, policy, etc., the fundamental scientist is sort of inhibited in terms of the kind of goals that, well, they're also unhinged, but we're also kind of uninhibited in terms of the kind of deliverables and goals and deadlines that we need to achieve. So what this means is that industry, when they have a new chemical that they're putting through their pipelines, they could look to, for example, basic microbiologists to do some of the experiments that they're not interested in doing in order to get this compound out there. And I think this is the power that academia has that really should be exploited. Just to give you a very specific example related to the work that we do, when we think about the future of epidemiology and surveillance, it's going to be sequence-based. We're going to move away from phenotypes whether we like it or not. We're going to move into artificial intelligence-based models that use sequence data to predict what the outcomes of an infection will be. This is not very far away. And so essentially, sequence data and making sense of sequence data is going to become the question if we're going to be able to convert this into products, right? And really what we find is that when you take sequences of drug-resistant strains, whether they come from clinics or from natural environments or from labs, what we do is this last step where we kind of curate and cherry pick, and we typically go with the mutations that we either know are associated with certain kinds of resistance or molecules that are associated with certain kinds of resistance. But there's probably a whole lot of background genetic variation in bacterial strains that simply does not get investigated because it may not be quote unquote important enough, right? Because it probably doesn't have high effect size mutations. What we find, though, in the lab, of course, is that epistasis is a major force that shapes the phenotype of drug-resistant strains, which means you can have mutations that have minor effects independently, but when you combine them with another mutation, you see these very large effect sizes, right? And so it may not be important for you to really deal with, the low-hanging fruit, but maybe some of these other, what we consider at the moment background mutations might actually be more important. So essentially, it's sort of the iceberg metaphor where we have a small number of sequence variants and mutations that we're all interested in, but there's this whole wealth of sequence information that's out there that nobody's going to look at. Most likely, industry is not going to touch. And so, for the basic microbiologists, this would be, I think, the way to go ahead. Finally, how do we bring about this kind of integration? Well, you would need a superstructure that would coordinate all of these different kinds of enterprises that are circling around the problem of AMR. There are major issues with setting up these superstructures. So for example, all of this, who sets it up? Is it a governmental organization, non-governmental, industry, academia? Who's sort of coordinating these efforts? More importantly, how do we find common vocabulary? So for example, what as a basic researcher I consider a resistant strain, a clinician may not, simply because it's not clinically relevant, but it's still resistant. So when I say resistant and when a clinician says resistant, are we really talking about the same thing or are we talking about different things? Chances are we're talking about very different things. And so we need to find common ground and figure out what we're saying, right, to each other. But more importantly, because we're working in all of these different spheres of research activity, whether it's basic science or the industry, we all have different sort of goals that need to be, benchmarks that need to be met, goals that need to be sort of ticked off. And without really disturbing these, because you do have very practical considerations of many things that all of us know, you still want to achieve a larger goal. And so how do you sort of compromise this at the local level without really changing systems too much, but leading to a larger kind of finding? And I think that these are all up for discussion, and we can talk about them or not, or think about them